So let us start today's lecture. So in today's lectures, we are going to discuss about body fluid compartments and their measurement method. So what are the most important objectives in this topic that I am going to discuss? The first part, the distribution of total body water in the body. Then what are the different ionic composition presence in different body compartments, and finally we will discuss about what are the different methods of which we can measure the different body fluid compartment. Before I proceed to the first objective, that is about the distribution of total body water, we must know about their exact location, right, where actually these body fluid compartments are exist, right? Then only we can discuss about their distribution pattern. So to know about the exact locations or their existence in body, you can see this diagram. Here you can see the left side, this is the dorsal body cavity, and the right side, it is the ventral body cavity. So if you see the structures in the dorsal body cavity, this cavity are mainly composed of two parts. One is the cranial cavity and another one is vertebral cavity. So the cranial cavity contains the brain, whereas the vertebral cavity contains spinal cord. Both the cranial cavity and vertebral cavity contain some special type of fluid called cerebrospinal fluid, and that cerebrospinal fluid acts as an body fluid presence in the dorsal part of the body, which is also known as the part of extracellular fluid, or it is also known as the transcellular fluid. If you see the ventral part of the body cavity, it is mainly composed of the thoracic cavity and abdominal cavity, right? So this thoracic cavity actually composed of pleural cavity, which is surrounding the lungs, and the pericardial cavity, which is surrounding the heart. At the centers, it is the mediastinum, right? So this part also contains some fluid called transcellular fluid, which is a part of extracellular body fluid, right? Now see, if you look into the cellular level, inside the cells there are certain types of fluids called intracellular fluids. At the same time, if you look into the outside of the cells, some fluids are there that surrounds the cells, or in between cells in a particular tissue, there are certain types of fluids called extracellular fluid. So it means the body fluid compartments are mainly the intracellular body fluid compartments and extracellular body fluid compartments, right? The most important types or parts of the extracellular body fluid compartments are, one is the blood plasma, and another one is interstitial body fluids. The interstitial fluid is the fluid which is present in between cells in a particular tissue. So other than plasma and interstitial body fluid, the transcellular fluid which is present in the body cavities also acts as a part of extracellular body fluid, right? But the intracellular body fluid is solely present inside the cell. Now why the measurements of all those body fluids is important? Because if you see the clinical link of these body fluids, you will find certain linkage, certain importance of the assessment of those body fluids. Say when a person loses blood or has edema, this changes occurs between plasma and interstitial fluid compartments. So there is two important clinical conditions, the loss of blood and another one is the edema, right? So in both the conditions affects the body fluid mainly body fluid compositions or body fluid compartments. So now we can say that the study of body fluid compartments and the measurements methods of body fluid compartments is very important in physiological point of view. Here you can see the intracellular fluid. It is mainly 2/3 of the total body waters. So we must know about the amount of total body waters, right? So if you see the total body water contents in healthy persons, it is around 60% of the total body weight, right? So 2/3 of that is total body waters and the extracellular fluids is 1/3 of the total body water, right? Now, so as I have already mentioned, these are the most important part of ECF. See here, the body fluid is important because it contains different types of molecules, and that molecules are essential for the functions of various physiological functions of cells. Now we should know what are the most important chemical compositions or ionic compositions present in body fluids, and where actually these different types of molecules or ions are present, and what is their concentrations or amount. So if you see the ionic distribution pattern in the left column and the right column, you will see certain difference. And the question is, these ionic compositions, how these ionic compositions in the body fluids are maintained. Now, to maintain the body fluid in a body fluid compartments, we must take some fluids as well as we also need to loss of that fluids to maintain the balance, right? So what are the major roles of water intake and water loss? So you can see in the left column. So for healthy person, the average intake of water per day, it is around 2.5 liter, right? Now question is, what are the major role of that water? So most of the part, around 60% of the body waters are getting from drinking water, right? And around 30% from food contents. At the same time, some of the body fluids are getting by the metabolisms of the body, right? So these all together, it is around 2.5 liters that we are getting per day on an average. So the maintaining of a certain level of body fluids is very essential. As we intake around 2.5 liters of waters per day, at the same time, to maintain the balance, we need to loss of that amount of waters too, right? So now question is, what are the major roles for the loss of that body water? So these are the major route, like around 60% of the body waters are lost by urine, right? And other than this, there are some insensible route, like the insensible route are the skin and the lungs. Both the insensible routes are used to lose a certain amounts of water, right? Other than this, some of the waters are lost with feces, around 4%, and some are lost through sweat. So here, if you see the average amount of loss of body water is also 2.5 liter per day. So it means the water gain and water loss, or body fluid intake and body fluid loss, is very vital to maintain the balance or homeostatic balance of a particular tissue or cells or even in organ systems. So these are the major routes of fluid intake and major routes of the water loss. Now, if you see the distribution pattern in different group of people, you will see a sharp difference here. So what are the most important factors that affects the total body water? The most important factors is age. Not only age, there is another important conditions that affects the body water, total body waters, is the body composition, right? Mainly the ratio of fat and lean tissue. So if you see some properties of the fat tissue, so fat tissues has a less capacity to captures the certain amounts of water in comparisons to the lean body mass, right? So that is why this is most important part, you can say, the total body compositions. Here, particularly the proportions of the fat and lean tissue are most important. Water is present mostly in the lean tissue, while fat contains very little water. So, individuals with more body fat have a lower percentage of total body waters. The most important point. Why? Because if you see the diagrams here, the infant on an average, if you see the total amount of body water is around 80% of this person's body weight, right? This is the maximum percentage. Why? Because in infant, in comparison to fat tissue, the lean mass is more, so that it will the infants can restore or stores more amount of body fluid or body water, which is followed by adults around 60%. In case of adult, you can see in comparison to fat mass, the muscle mass is more, or lean body mass is more in adult male. So that is why the total body water percentage in adult male is little higher in comparison to adult female, right? So why there is a less body water percentage in females? Because the fat tissues in female individuals is much more in comparison to muscle mass, right? At the same time, you can see the old age persons. In that particular case, total body water percentage is around 45%, because with increasing age, there is an increase in the loss of muscle mass, but getting more and more amount of body fat. That is why the old age persons also have less percentage of body waters. So from this slide, we can say the maintaining of total amount of body water or total body water percentage is depend on certain factors, mainly the age factors, as well as body fat percentage and lean body mass. Now, as I've already mentioned that body fluids are mainly divided into different compartments. Some fluids are present in extracellular part and some are present in intracellular part. Now we'll see what is the exact percentage of different types of body fluids in different body fluid compartments. You can see these parts, as I've already mentioned, that extracellular body fluid, it is divided into 3 different compartments: the plasma, then interstitial body fluid, and transcellular fluids. So you can see from this diagram, this is the blood vessels which contain blood plasma, and this is the space in between cells called extracellular space, and that extracellular space contains the interstitial body fluid, right? And which is not shown here, transcellular fluid, which is basically presence in specific type of body cavities, right? Now, if you see the total amount or percentage of body fluids in different compartments, you can see from these diagrams, it is around 42 liters. If you see the body fluid percentage or body water percentage in healthy persons whose age is, weight is 70 kg, right? So for that persons, the total volume or total body waters is around 60%, which is around 42 liter, right? And that 42 liters is divided into 2 compartments. So here you can see it is 28 liters presence in intracellular fluid compartments. Means intracellular fluid compartments means, yes, it is all about the inside the body fluid, inside the cell. And 14% presence in the extracellular compartments. That means around 2/3 of the total volume are present in intracellular compartments, and 1/3 is present in extracellular fluid compartment, right? Now, if you see the extracellular fluid compartments, which contains the total of 14 liters, and that 14 liters is divided into different part. Like the most important one is called the plasma, which contains 1/4 of the extracellular fluid volume, right? And in extravascular fluid means the fluid which is present in interstitial space. It is 3/4 of total extracellular fluid volume. And here extravascular fluids again divided into two parts. One is interstitial and one is transcellular. So this chart actually gives us a clear picture about the percentage distributions of body fluid in different body fluid compartments, which is very essential to know, because we know that the maintaining of body fluids in a specific compartment is very essential for physiological functions. So at the same time, as it is physiologically important, so we must know about their concentration, about their volume or amount presence in different fluid compartments. Now see, since this body fluid is important for physiological point of view, the regulations of water is very vital. The question is, what are the most important points that involves the regulations of body water intake, right? Yes, there is certain physiological state, and that physiological state influence the intake of body water. The most important one, you can see the decline in plasma volume of around 10 to 15%. Just now we have seen the total amount or total volume of plasma fluids around 3 liter, right? So if there is a decline of around 10 to 15% of that plasma volume, it influence a specific site presence in hypothalamus called thirst center, right? And that activations of that thirst centers stimulates the water intake, right? So it means you can say the decline in plasma volumes is an important indicators or important regulators of that mechanism, regulatory mechanism. Another important part is increase in plasma osmolarity. So plasma osmolarity means the concentrations of particular solute present in blood plasma, like the concentration of sodium ion or concentration of potassium ion, right? Or concentrations of other substance. So here the plasma osmolarity is an important point which involves the regulations of water intake. So what is this? Increase in plasma osmolarity. It means so many solutes in blood plasma. So this is again another important conditions. Other than this, there are some other pathway that also activates the hypothalamic thirst centers for the regulations of water intake, right? You can see from this diagram, there is a condition called hypohydration. In hypohydration, it means the water levels in cells is less than optimum, right? So it means the concentrations is solute concentration is more, or concentrations of particular solutes in that fluids is more, right? So it means there is a changes in the blood osmolarity, right? So increase in blood osmolarity stimulates the receptor presence in hypothalamus, that is the osmoreceptors, and activations of that osmoreceptors ultimately acts in two different paths. One is the stimulations of the thirst centers, another is the activations or stimulations of the pituitary to release a certain types of hormones called ADH or vasopressins. So here ADH played an important role in the tubular water reabsorptions, and by the process of tubular water reabsorptions, it maintains the body fluid. At the same time, by stimulating the thirst centers, it increases the water intakes. Both the conditions ultimately helps to restore the blood osmolality, that is around 295 to 300 milliosmol per kg water. So this is one important mechanisms that is used to maintain or regulates the water intake. The first one is The conditions called decline plasma volume is around 10 to 15%, and then changes in the plasma osmolality, mainly increase in plasma osmolality, and there is some other pathway like baroreceptor pathway and the role of angiotensin II. Both the conditions also involves the regulations of water intake. Now see, to maintain the fluid balance in cell, there are some physiological phenomena works in proper way. So what are those physiological phenomena that involves to maintain the fluid balance between cell? So these are some important points called the semi-permeable membrane. We know the most important features of semi-permeable membranes. It allows water molecules, right, freely. So there is a free passage for water molecules through cell membrane. But if you see the characteristics of some cell membranes, it obstruct the entry or exit of certain types of solutes or other ions or other molecules freely, right? Now, due to that obstructions of solute movement, the inside of the cell membranes and outside of the cell membrane, the concentrations of solute differs, right? And that difference in solute concentrations in two different sides of cell membrane creates or generates a force called net osmotic force, right? So what is the net osmotic force, or what is the most important features of that net osmotic force? It determines the direction and magnitude of water movements across the biological membrane. So water movements is essential for the cell, but for how long the water will move from one side to other, that depends on the equilibrium conditions. Until and unless there is no equilibrium forms between inside and outside of the cells, water will move. But due to the after the equilibrium in both the sides, so the force called net osmotic force prevents the further movements of water molecules through the cell membrane, right? So this prevention of further movements of water molecules is essential to maintain the fluid balance inside the cell. This is where the importance of net osmotic force or generations of net osmotic force. So here you can see this force arises from difference in solute concentrations between two compartments, and it ensures fluid balance within and between cells. You just see the initial state. In the initial stage, you can see in one side of the cell membrane, the number of solute is very less in comparison to others. So to maintain an equilibrium state in both the phases, what it needs the movements of water to maintain the concentration of solute in both the sides, right? So that movement of waters will continue until the equilibrium in both the sides. After that, this net osmotic force or osmotic pressures prevents the further movements of solvent here. Now, as I have mentioned that the ionic compositions or ions are very vital for different type of cellular functions or physiological functions. So if you look into the table, two different group of ions are present in different body fluid compartments, mainly the cations and anions. Here we will see what are the most important cations and most important anions. You must remember the name and list, right? Because it is very vital in clinical aspect, right? So what are the most important cations and where actually these cations are found? You can see the most important types of cations are sodium ion, potassium, calcium ion, magnesium ion, and some other substances. So sodium, potassium, calcium, magnesium, these are cation, right? And see what are the anions? Chloride ions, bicarbonate, phosphate. Other than this, proteins also present in body fluid compartments. Now we will see about their distribution pattern, right? If you see this diagram or this graph, all those cation and anion are distributed in both the extracellular fluid compartments as well as intracellular fluid compartments, right? But the concentrations of those ionic distributions are not same in both the extracellular fluid and intracellular fluid. Now we'll see in this diagram, you see the upper portions, sodium ion, which is a cation, and the lower portions, the potassium ion, which is also cation. So the most important or maximum amount of cation present in extracellular fluid is the sodium ion. Remember, the maximum amount of cation present in extracellular fluid is sodium ion, whereas the maximum amount of cation present in intracellular fluid is the potassium ion. So what is the average amount of both the sodium and potassium present in extracellular and intracellular fluid? You can see here extracellular fluid means the fluid present in blood plasma, fluid present in interstitial space, fluid presence in transcellular fluid. Now if you see the amount presence in blood plasma, mean in extracellular fluid, you see it is around 142 millimoles per liter, right? Whereas you see the potassium ions, this one in this part, potassium ions, this skeletal muscle cell. So inside the muscle cell means intracellular fluid, it is around 140 millimoles per liter, right? So this is used to just know about the distribution patterns of the cation here. Now if you see what are the other things, like if you see the extracellular fluid contains a maximum amount of cations is the chloride ion, whereas in intracellular fluids the maximum amount of anions is the phosphate and some inorganic ions, right? So this is the general distribution patterns of some important cations and some important anions in both the extracellular fluid and intracellular fluid. Other than this, some other cation and anions are also equally distributed in both the extracellular fluid as well as intracellular fluid, which is very essential for cellular function. Now see, as I have already mentioned that between cell or between blood vessels and interstitial fluid, there is a certain types of membrane, right? And that membranes are very specific, that doesn't allow all the different types of molecules. It is selectively permeable, right? But for the cellular functions in different point of time, it needs the movement of different type of molecules to maintain the cellular balance or fluid balance in cell or maintaining cellular functional integrity. Now here we will see how the different types of molecules moves from intracellular fluid to interstitial space, from interstitial space to inside the cells, or from blood capillaries to the interstitial space. Here you can see there is a specific law that acts here during the movement of different type of molecules from one fluid compartments to the other, right? That is called a Starling force. So what is the most important features of the Starling force? Two different pressures helps to maintain the fluid balance between intra and extracellular fluid compartments. So one force is called hydrostatic force, another one is osmotic force. See, Starling force describes how fluid moves across the capillary wall between the blood plasma and the interstitial fluid. That is totally based on the balance of hydrostatic and osmotic pressure. See, suppose this is a blood capillary, right? And this is the blood capillary membrane. The outside the blood capillary membrane, there is an interstitial fluid. But inside the blood capillary, there is a plasma. So in different point of time, it needs the movement of molecules from blood plasma to interstitial space. Now, how these movements of molecules are actually maintained? So to maintain that movements or to maintain the fluid balance inside plasma as well as the interstitial space this stalling force act. So you can see here in this part, see, suppose this is the blood capillary, right? And there is a capillary membrane or capillary wall, right? So how this capillary wall acts here, a specific pressure called hydrostatic pressure pushes water out of the blood capillaries, while osmotic pressure generated by plasma proteins pulls water back in the plasma fluid, right? So one actually push and another is involved the pulling, right? And pushing and pulling is both the mechanisms acts in a conjugated way, right? So that helps to maintain the balance of fluids in both the interstitial space as well as the plasma, blood capillaries. You can see here the balance between these force maintains normal fluid exchange between plasma and interstitial compartments, whereas here hydrostatic pressures and one is osmotic pressures. So what is the meaning of the term hydrostatic pressures? Actually, in case of this particular points, if there is any concentration difference between blood plasma and interstitial space, so the molecules will move, mainly the water molecules will move from higher concentration gradients to the lower concentration gradient, right? And it will continue until and unless it maintains the equilibrium in both the conditions, means in both the interstitial fluid as well as the blood plasma. And another one is osmotic pressures. That osmotic pressures actually regulates or determines the movements as well as the magnitude of movements of the molecules. So here these two mechanisms has to maintain the fluid balance in interstitial and this blood and plasma. But if you see the water movements between intracellular and extracellular compartment, means inside cell and the outside cell. Here outside cell means it may be interstitial, it may be blood plasma, or it may be transcellular fluid, right? So that depends on osmotic gradient that is created by electrolytes only, right? No hydrostatic pressures works, but instead there is an osmotic gradient, and osmotic gradients actually means the concentration difference of a particular solute in two different compartments of body fluid compartment, right? When the extracellular fluid has the same osmolarity as the fluid inside cells, water doesn't move in or out of the cells, so that intracellular fluid volumes stay the same. So now question is, this is very important point, how the intracellular fluid volumes are maintained in a normal or optimum level. So this is totally depends on the osmolarity. If the osmolarity in both the outside of the cell as well as the inside of the cells is same, it means the concentration of a particular solute in inside of the cell and concentration of same solute in outside of the cells is same. So in that particular stage, there will be no movement of water through the cell membrane. So by these mechanisms, so intracellular fluid volumes are maintained, right? So this is how the fluid movements occurs in different compartment present in extracellular part, and this is how the movements of water molecules takes place between extracellular and intracellular compartment. See, most important point here, how do we measure the plasma osmolarity, and how this plasma osmolarity measurement defines that it is the osmolarity of ECF, right? So as you already know, the ECF consists of three compartments. I am repeated the terms: the interstitial fluid compartments, then plasma, then transcellular fluid. So here question is, how the measurement of plasma osmolarity defines the osmolarity of extracellular fluid. The most important points behind this is it is very easy to determine the plasma osmolarity in comparison to the determine of the osmolarity of other compartments like osmolarity of interstitial body fluid or osmolarity of transcellular fluid. Right? This is first point. What are the others? See, since plasma is the only ECF compartments that can be easily sampled, its osmolarity accurately reflects the solute concentrations of entire ECF, right? So here you can take one example to understand this. The major contributors to plasma osmolarity, like sodium ion, it is a solute, right? And its associated anions, mainly the chloride and bicarbonate, along with some contributions from some other molecules like glucose and urea. So here, as it is easy to sample, I mean the plasma samples easy to take. So the test using this plasma sample actually determines the overall osmolarity, overall concentration of solute presence in extracellular compartments. That is why most of the time to know about the osmolarity of extracellular fluids, we measure only the plasma osmolarity. Remember, to know about the osmolarity of extracellular fluid means the concentration of solute present in extracellular fluid, we only determines the solute presence in blood plasma by taking the sample from blood capillaries or veins. So therefore, any changes in plasma osmolarity, such as there are certain conditions such as dehydrations, overhydrations or electrolyte balance, represents a change in osmotic conditions of the entire extracellular fluid, which in turn influence the water movements between ECF and ICFs to maintain the osmotic balance. So here the maintaining osmotic balance is very vital, right? So how do we know that whether there is a difference in osmolarity presence in body fluids or not? That is only determined by knowing about the concentration of solute presence in different compartment, right? So here this measurements of plasma osmolarity actually help us to know about the concentration of solutes as well as the molecular movements between intra and extracellular fluid, also help us to know the osmolarity of intracellular fluid as well as the extracellular fluid. So if you see the average osmolarity of sodium ions, that is around 270 to 290 milliosmoles, that needs to maintain in the cell. So the maintaining that osmolarity, how it is maintained? By hydrostatic, by osmotic pressure, right? See, an important conditions called disorder of water balance or dehydrations. So what actually happens in dehydrations, or how, so why dehydrations occurs? These are certain routes through which there is a high chances of water loss, and that water loss is an important phenomena by which dehydrations may occur, right? So dehydrations occurs due to excessive loss of water from the extracellular fluid, So what are the major routes? Sweating, diarrhea and inadequate intake of water. Now, as water is lost, you can see from this diagram here, as water is lost from extracellular fluid, water loss from extracellular fluid means it affects the osmolarity of extracellular fluid, right? So the extracellular fluids will be more concentrated, right, in comparison to the inside of the cell, right? Now, as the extracellular fluid is more concentrated, now to maintain the balance between the extracellular fluid and intracellular fluid, the cell will start, the water molecules will move from inside to the extracellular fluid to maintain the concentration of a particular solute presence in that extracellular fluid, right? So due to the movements of water molecules from cell to the extracellular place, there is a high chances the shrinkage of the cell, right? So this is what is the disorders of water balance. So there is a shrinkage of the cells due to the dehydrations or due to the disorders of water balance. And most important mechanisms access the changes in the concentration of solute in extracellular fluid. Here, this part, and due to that changes of osmolarity, the water molecules move from inside to the outside, right? Now, the measurement of extracellular fluid or the fluid compartments is very vital. So what is the mechanisms by which you can measure the body fluid compartments? Yes, there is a specific methods called indicator dilution method, right? That indicator dilution method actually acts using the principle of conservation of mass, right? The conservation of mass principles, the principle which is used to measure the body fluid compartments. So before proceed to that methods, we must know what are the important points that we should maintain during the measurements of body fluid compartments. There is suitable marker or dye that is used in indicator dilution method, that is used for measuring body fluid compartments, and it must fulfill certain essential criteria. So a specific type of dye are used which act as an indicators. So it should maintain certain criteria. It should mix evenly throughout the compartments to ensure accurate measurement. Suppose we want to measure the extracellular fluid compartment. So before using that particular dye into the extracellular fluid compartment, we have to ensure that this dye should evenly mix with that fluid presence in the extracellular compartment, right? Dye must be non-toxic. If it is toxic, it will alters the cellular behavior. So before using that dye, we have to check whether it is toxic materials or non-toxic, and it should be physiologically inactive, so it doesn't alters normal body functions. The dye should not affect the distributions of waters and other substances in the body. So once we use that particular dye by injecting, suppose we are injecting that particular dye into the vein, right? So there is a high chances once the dyes are injected, there is a high chances of alterations of the chemical compositions present in the that fluid. So we have to we have to take some precautionary measurements before using that dye. It must remain chemically stable during the experiments, or any changes in its amount should be known. What amount of dye we are using for that particular experiment purpose, we have to measure the amount before using, right? So these are some important criteria that we should follow whenever we are using the dye for the measurement of body fluid compartments, right? Now let me show you how actually these body fluid compartments are measured. Suppose for the measurements of blood plasma, a specific type of dye are used, but the same dye are not used for the measurement of body fluid presence in interstitial space. At the same time, for the measurement of body fluid presence in intracellular space is also different. It means there are mainly 3 different group of dye or indicator are used for the measurement of body fluid compartments. We will see what are those dye. Now, what is the exact methods or how these indicator dilution methods are act? Suppose Suppose in this test tube, we have a known volume, right? Known volume of fluid, and we are using a certain amount of dye or indicators whose mass is the known mass, that is m, right? Now if we want to know about the concentrations of that, it is actually a particular solute, suppose, right? And the mass of that solute is known, and the volume also is known volumes. Now if we want to calculate the concentrations of that solute presence in this particular fluid, how to calculate? That is, this is the formula how to calculate the concentration of that solute present in this tube, because we already know about the volume of that particular fluid presence in this tube. We know the mass of the substance or indicator or dye used in this tube. Now using the value of both the volume 1 and the mass, we can calculate the concentration of the dye used in this tube, right? Now suppose the solution which contains the dye as well as the fluid, right? Now that whole solutions are transferred to the second tube, right? Before transfer that fluid to this tube, and it also contains certain amount of fluid, but that fluid is unknown fluid. This is unknown, right? And the same fluid are used in the second tube. Clear? So whatever the amount of things are used, added in the second tube, in this second tube also contains type of dye, right? At the same time, not only the same type of dye, the mass of the dye also is same. So it means in the second tube also we know the mass of the indicator, mass of the dye, mass of the substance, right? So it is known, but it is unknown, right? Now here if you want to know the concentration of that solution, what will be the formula? So again the same thing, that is the concentration is equals to mass by volume. Clear? It means this is known for us because we used the previous solutions, right? Where the mass was known, but it is not known. Now it means here it is M2, suppose this is M1. See, in both the case, this is the case 1 and this is case 2. In case 2 and case 1, what is same? The things that is equals is the mass, right? So here M1 is equals to M2 from case 1 and case 2. It means C1 V1 is equals to C2 V2, right? What is our intentions? Our intentions is to know about the volume present in blood capillary or volume present in extracellular fluid. Clear? Suppose we want to measure the plasma volume presence in blood capillary. So what is the method? That is the dye dilution method, right? Now here this is known, concentration 1 known, V1 is also known. Now we have estimated the concentration of that particular dye presence in second tube. It means how in lab we easily can determine the concentration of a particular solution where the mass is known, right? If we take a certain amount of samples from this tube in lab, so easily can determine the concentrations. So it means the concentration here is known. We already determined that one, right? So what would be the volume? That is V2 is equals to C1 V1 by C2, right? So now C1 is already known, V1 also known, and C2 just determined from this tube. Now we easily can determine the volume of this tube, right? Here, law of conservation of mass means the dye that was used for the experiment purpose, the mass of that dye was not changed. It remains same in this condition and in this condition, right? The same principles is generally used to measure the body fluid compartment or fluid presence in different compartments of body, right? So suppose this is the blood and capillary. Now we want to measure the plasma volume, right? So for that, suppose here certain amount of dye are used, and that dye is known dye, and mass is also known, and that is injected into the vein, right? That is injected into the vein. That is injected into the vein, right? This is vein here. Clear? Now, before injecting, we already know that one, and we also know the V1, and we have calculated the C1, right? Now, from that, a certain amount of samples are collected, right? And after collecting, we have calculated the C2 or concentration of that dye presence in this blood plasma. So after knowing the concentrations, we have used the value into this formula, right? So by this way, we can measure the volume of a certain compartment or body compartment. This is what is the dye indicator method. Now, let me show some other important point. As I've already mentioned that there are different types of indicators are used. So let me show what are the other indicators. So for the determinations of plasma, the most important type of indicators is Evans blue, right? Radioiodinated fibrinogen, then albumin. The most important one is Evans blue, that is used to determine the blood plasma. Why these are specific? These are very specific because as you've already seen that in the this dye are not allowed through the capillary one, so that there is a less chance or no chance of leaking those dye from blood plasma to the interstitial fluid. So whatever the value will get or volume will get, that is the total volume of blood plasma only. On the other hand, for the measurements of ECF volume, so these are important type like sucrose, inulin, mannitol and sodium chloride. The features of this is here extracellular fluid volume. Actually it means the determination of what? Interstitial fluid volume, right? So that is also very specific. If you use this type of dye, that dye easily can cross the capillary wall to the interstitial fluid, right? Up to interstitial fluid, but it doesn't allow to move that dye to the inside of the cell. So whatever the volume will get by using this type of dye, that will be sticked into the extracellular fluid volume only, not intracellular fluid. So to know about the volume or intracellular fluid volume, these are the different dye called heavy water, tritiated waters, aminopyrines and antipyrines. These are important dye, and similar to the previous case. So if you want to know the total body waters, if you use this type of dye, they can cross the cell membrane, right? They can distribute into the extracellular fluid, as well as they can present in the capillary systems. So it means using this dye, if it determines the fluid volume, it actually determines the total body water, right? But we cannot determine directly the indirect body, intracellular body fluid. So to determine the intracellular body fluids, we have to know about the value of other indicators that is determined by direct method, like extracellular body fluid as well as the plasma fluid. So if we subtract the value of extracellular body fluid from total body water, it will give us the value of volume present in intracellular site or intracellular fluid volume. Clear? At the same time, it is also difficult to determine the interstitial fluid directly, but we have determined the plasma volume. We also determine the total body water. If we subtract the plasma volume from total body waters, we can guess the value of volume presence in interstitial fluid. So this is how this dye dilution method is used for the determination of a volume presence in different body fluid compartment. Thank you.